As mankind continues into the 21st century, one technology will be instrumental in helping us to gain new perspectives on the world around us. From the environment we inhabit into the very cells of our body, the science of sensors is set to revolutionize how we live our lives.
The major benefits coming out of the sort of scientific work we do, both societal and economic. From a societal perspective, it means we'll affect the quality of people's lives into the future, from the very young to the very old.
Irish investigators are united in an effort to develop innovative and novel applications that will, in the near future, radically alter our relationship to our surroundings in terms of communication, interaction, and human health.
I think sensors will have a dramatic effect on our society in the near future. The applications, as I say, are limitless, and we're seeing sensor technology being applied more and more in different areas that affect people's lives. It can be from environmental monitoring to biomedical monitoring that we carry out here. So sensors will have a huge effect on our lives in the future.
Sensors are already playing an important but mostly functional role in the lives that we lead. We've become used to the way that they help to regulate our lives. Barry Smith is an Irish investigator based in UCD. He's working to usher in the next generation of intelligent sensors, sensors which will anticipate, process and respond to our unique personal preferences.
Sensors are the eyes and ears of computers. They allow us to connect the physical world in which we live, the world in which we work and play, with the digital world of the internet. The big development that's been happening is that sensors are becoming cheaper and they're becoming connected, and mobile phones are playing a key role in this new vision. These devices are no longer just telephones, they're information devices. We use them to browse the internet, we use them to store our photographs, we even use them to watch videos. Sensor technology allows us to take all of this rich multimedia information and make sense of it, so that that information can be personalized and made truly useful for the individual.
Barry Smith is taking advantage of Ireland's increasing dependence on the mobile phone. He's developing software for use on the phone, which will build and tailor unique information based on our personal preferences from the items that we buy, the music that we listen to, and the places that we visit. Combining these personal preferences with other systems, such as GPS, has the potential to transform our lives and help us to get the kind of information we need when and where we need it.
So in this example, Jill has just left the tourist office and her mobile phone has been loaded up with maps of Dublin City, and she's about to embark on a personalised tour of Dublin. Her mobile phone is using location sensing technology combined with information about Jill's preferences and her interests to produce for her a personalised tour around Dublin. It'll focus in on the museums, the art galleries, the exhibits that she might like to visit on her day. This technology presents some very significant opportunities for the advertising and retail sector. It allows stores to advertise their wares in a much more targeted fashion. It allows individuals to shop much more efficiently, and it allows those who are looking for a bargain to hunt out that bargain much more effectively.
In its commercial application, the idea is that shops could register their content with a particular location. When a user with internet access moves through the location, the software on the phone would be able to pick up the content that has been registered by the shop. This content can then be compared to the user's preferences, and if there's a match, they'll get a message on their phones to tell them about it.
So in this example, Jill's mobile phone has learned about her preferences and her interests. And as she wanders around Dublin City enjoying the sights and sounds, as she goes by particular locations, she's alerted to particular pieces of information associated with that location that may match her interests or preferences. As she walks by the Kilkenny shop, she's alerted to the fact that there's some Waterford crystal on offer that she may want to bring back home with her.
While this technology undoubtedly has its advantages, it also presents certain challenges.
One of the big challenges in this area of research is the privacy implications of monitoring people and using sensors to learn about their preferences. And it's all about getting the balance right. What can we learn about people, about their needs, and what sort of service can we provide on the basis of what we've learned? Sensors can also change the way that we manage and monitor personal health care. We can now communicate information about our physical well-being directly back to our physicians so that they can keep in contact with how we're responding to various different treatments.
Sensor technology will have a huge impact on our everyday lives. Simple monitoring of chemicals in the body and transferring that information back to data banks so the information can be processed and maybe we can actually look at disease states before they occur. So we can actually monitor people as they go about their normal daily lives and get information that will help improve their lives and predict maybe that disease states that might occur in the future.
As sensors become ever more sophisticated, investigators are working on ever smaller devices with a new destination, the human body. John Lowry of NUI Maynooth is looking at monitoring important chemicals at work in the most complex part of the human body, the brain. The brain is a giant messaging system that controls everything, from our thoughts and emotions to our physical movements. It's made up of billions of nerve cells or neurons. These neurons send and receive messages from the rest of the body using brain chemicals called neurotransmitters. In disorders like schizophrenia, the neurotransmitters get blocked and the signal becomes weak. John's sensors measure the level of these chemicals and record real-time brain activity, giving us for the first time a unique insight into the interplay of the brain's chemicals.
What we have here is a sample of brain fluid, and this is the analysis of that sample. So basically we have these peaks which represent different chemicals in the brain. So we can measure the height of these peaks and the time that these peaks appear on the graph, and that tells us which particular chemical we're actually monitoring in the brain and the concentration of that chemical. It's a fantastic technique in that we can get, you can see several peaks on the screen, so we can get information on several chemicals from the same sample. The big disadvantage really is that this is a sample that might have been collected several days ago. So we're really looking back in hindsight. So what we want to do is to be able to look at the behavior and relate the behavior directly to the chemical changes in real time. And we also want to be able to look at, different chemicals changing. So it'd be nice to be able to look at glutamate, dopamine, glucose, lactate, all changing together in real time. Okay, what we have here is a typical sensor, and basically in terms of size, if you were to take a hair from your head and put it alongside this sensor here, their dimensions will be exactly the same, about 100 microns in diameter, or 1,000th of a millimeter. The active part of the sensor is really the tip of the sensor up towards the top. And basically we modify the surface of the sensor so that it's selective for a particular chemical. So for example, if we want to measure glucose, we modify the chemistry on the top of the sensor so that it will actually measure glucose alone and no other chemical. So we have a wide range of different sensors that have been specifically developed for a particular chemical. And our focus has been on chemicals that are implicated in different disease states in the brain.
Using the tiny sensors, John has set up an artificial environment that simulates the interaction of chemicals in the brain. These are the first steps in a process that may result in real-time recording in a human.
What we have here in the glass cell, we're making a copy of the brain environment. So we have a solution in here which mimics the brain extracellular fluid, in other words, the fluid that's inside the brain. This instrumentation here, the clips on that, are connected to our sensor, which is placed in the solution. What we do then is we change the concentration of oxygen in the fluid. And when we change the concentration of oxygen in the fluid, the sensor is able to measure those concentration changes, and we are able to record on the computer screen. So now we're actually looking at the chemical changes in real time as they occur in the brain. Well, the big advantage, for example, if you take schizophrenia, there are several chemicals assumed to be responsible for schizophrenia. Typically, those chemicals are dopamine, glutamate, and serotonin. A lot of the drugs for diseases like schizophrenia are based on changing the concentrations of one or other of those chemicals. But what we don't know yet is the interplay between those chemicals and how that interplay creates a disease state. So by using the sensor technology, we should be able to measure each of those chemicals in real time and understand the complex relationship between those chemicals and that devastating disease state which affects so many people. The collaborations in Oxford now span back about 10 years when I worked here as a postdoctoral fellow here in physiology, and I've maintained the contacts with several academics in Oxford. It's a wonderful place to come and visit, very stimulating, and people are very, very interested in collaborating and working together. It's a wonderful opportunity to link NUI Maynooth with University of Oxford and carry out this type of research.
John is collaborating with one of the world's leading neuropsychologists, Professor Nick Rawlins, who works specifically in learning and memory function in the brain. In the same way as chemicals in the brain change levels when a person is suffering from schizophrenia, this also happens when we do a specific task for the first time and subsequently form a memory. For the first time, John's sensors will provide information on the chemistry of memory processing.
What we want to do is to go beyond just which bit of brain seems to be essential for you to be able to do this sort of task, and to ask, well, let's look at it another way round. When is a particular bit of the brain engaged? When is it activated? What turns it on? And that's, of course, what you can do with these sensors.
Using Lowry's sensors, Rawlins and his team look at the behaviour of rodents.
Basically, the animal carries out a memory task, for example, on a maze, and it has to remember where it was going and carrying out that task on the maze. What we do is we use the sensors to look at how the chemicals in the brain are changing as the animal actually carries out that task. In other words, how it remembers specific areas of the maze that it has to go to.
We can tell when particular bits of this animal's brain would be activated. And so if we're trying to figure out how he remembers where he's gone, or how he decides where to go to next, what we want to know is which bits of brain are activated, involved in that decision. And so the goal is that he can run around doing things, going about his life, looking for food, keeping track of what he's been up to, and then these signals should tell us which bits of brain are involved. And they'll tell us when they're involved, and ultimately they might even tell us how they're involved. That's the goal.
We're actually getting real-time information from the sensors as to how these memories are formed or what chemicals are changing during the memory storing process. And that's never been done before.
10 years ago, after the animal had gone through an action lots and lots of times, you could subsequently look in the brain and see which bits appear to have been active. That's very different from moment to moment, second to second. A decision is made. Where was it made? What was active in order for it to be made? So, I mean, that's an extraordinary thing to be able to do.
Basically, the next step is to take this technology into the clinical or patient environment. We've actually modified the technology already, the sensor technology, so that we can actually implant it in patients. Really, for us now, the next step will be to make contact with a neurosurgeon, for example, who's willing to apply the technology in, for example, head trauma or epileptic operations.
For Lowry and his sensors, this is just the beginning. With real-time monitoring of brain chemicals, clinicians and neurosurgeons will have real markers to identify and treat a whole range of diseases.
This is a bit of the brain that goes radically wrong in Alzheimer's disease. What is it about it and about the way it normally works that results in it behaving abnormally sometimes? If you don't know what the mechanisms are inside, you can't get the answers to that. Give us the mechanisms and we can start looking for the answers. And if we have the answers, then of course we can try to respond with therapies.
Diagnosing disease at an early stage and providing guidance for suitable treatment is vital for the health of individuals. One Irish team of investigators is tackling Ireland's and the world's #1 biggest killer, heart disease.
On the 8th of June, on a Sunday, I was at home and got some chest pains initially. I decided to go to the A&E in Beaumont. By the time I arrived in here, I was actually in a lot of discomfort. And the next day I was diagnosed as a heart attack. Every 2 years, for about 10 years now, I've gone for a regular health check. And I'd had a check last autumn. It had shown that I was in good working order and hadn't given me any cause for concern. But that just shows how this can come up on you, and that there are many different reasons, including ones that perhaps are not as well known as others, why people have heart attacks.
Professor Dermot Kenny of the Royal College of Surgeons is a specialist in the area of heart disease.
What we have here are coronary angiograms. And what happens is a little tube is put into an artery, normally in the groin, and then threaded up into the heart. You see this little tube in here, and it's injecting dye into the blood vessels of the heart. There's an abrupt cut-off here. So this is stopped, and this patient has just had a heart attack. If you look over here, this is after the patient has had an angioplasty, and you can see that the dye is spreading relatively normally. So the blood supply has been restored, the blockage has been unblocked. When you look and you can see that there's a blockage here in this vessel, that blockage is due to two things, a combination of two things. One is that the blood vessel is damaged, and the second, the blood stops flowing. What we're interested in understanding is how can we predict why that blood will stop flowing.
As part of a unique early prediction system for heart patients, Professor Kenny is using novel biochip technology developed by researchers at DCU's Biomedical Diagnostics Institute, led by Professor Brian McCrath.
Well, the technology we develop will directly affect the quality of people's lives. That is, doctors in clinics and in accident emergency will make decisions using our devices. That's a huge motivation for all of us. Yeah, we're very fortunate that the body itself is kind to us in providing us with early messengers of disease itself. The various diseases will give off biomarkers, molecules that are early indicators of the disease itself. And heretofore, it was very difficult to measure these biomarkers at very low concentrations. But now, by bringing together the various technologies that we have at our disposal, like nanotechnology, photonic technology, microsystems, lab on a chip, we can bring all these together to get a really exquisite sensitivity to measure these molecules at the lowest possible concentrations that give us a very early indication of the presence of the disease itself. And of course, that's very good news for the patient.
The BDI team is producing innovative, intelligent chips that can mimic the conditions experienced by the blood flowing around the body and can then identify the first sign of problems, giving clinicians a vital picture on progressive heart disease. One of Kenny's key projects is an investigation into the behaviour of blood platelets. Blood platelets play a vital role in the regulation of blood flow. Platelets are tiny microparticles that exist in low concentration in the bloodstream. Usually they are unactivated and move smoothly through the vessels, but sometimes they become activated and get sticky. When this happens, they join together to form clots which affect the blood flow. The resulting clots can cause stroke or can lead to a heart attack. The aim of clinicians is to monitor the activity of these blood platelets and thin the blood with drugs when these clots start forming, helping smooth out the blood flow.
Previously, very little was known about the degree of activation of these in the blood. Now here at the BDI, we've developed a new device which can determine the degree of activation from a small sample of blood itself, which we take from the patient and put on a chip itself. So this is a biochip, an example of what we call lab on a chip technology, where we take all the functions you would typically find in a laboratory like this here, sampling, mixing, detection, separations, and shrink it all down to a single chip in a disposable piece of plastic. And this is exactly the chip we use in the Platelet project.
Clinicians need to study how smoothly the blood platelets are running through the coronary vessels of patients with heart disease. The BDI's platelet chip can identify if rogue sticky platelets are likely to clot. giving clinicians the opportunity to administer drugs that will thin the blood and help distribute it more easily around the body.
But essentially, inside this little biochip device, we flow blood along a surface which mimics the inside of the vascular system. So we coat that surface with proteins which are typical of what you find in the vascular system. And then we measure the degree to which the platelets interact with those proteins, essentially how sticky they are, how activated or inactivated they are. If they're activated, they become very sticky. And it's a bit like the analogy of a person walking in muddy boots across a muddy field. It slows them down. We simply measure the rolling speed of those platelets across the surface, and we can use the distinction of speed of the rolling platelets to determine the degree of activation or inactivation.
So this is the image that I see when I look down the microscope. When we get a blood sample in, we incubate it for about 10 minutes with a special dye that stains the platelets, so that when we shine a certain coloured light, this is what we see. The platelets start to fluoresce.
The results that we can get from this chip can occur in seconds from very, very small amounts of blood. Up until now, an experiment like this would have taken us approximately 2 days. An interpretation of that data would have taken us another 2 days. Where we're going is to a device like this over here, where we'd be able to put the chip and get information out in seconds. I mean, the goal in medicine always is to prevent. What we don't want is we don't want the patient presenting with a heart attack. Ideally, we get out there and we screen patients, so we'd like to see this in every GP surgery.
The activity of the platelets in blood is something that I can't really do a lot about without the benefit of medical science, without the benefit of ongoing medicine.
Thank you. How's the rehab going?
It's going quite well, yeah.
Genuinely?
Yeah, no, it is going well. I mean, I was saying to Gail before. Therefore, it's important that I know and that ultimately the medical people are able to give me the direction as to how much of an issue or not the platelets are. So this project will help them to identify that. And in the not too distant future, I'd like to think that I could go to my GP and I'd do a simple test, which would allow him or her to say to me, look, this is where your platelets are at and your medicine needs to be changed accordingly. So it should make my ability to prevent something like this happening again much easier.
The success of this lab-on-a-chip technology offers enormous potential for preventative healthcare in developing countries.
Over 6 million people die every year in the developing world from diseases which, if detected early enough, could be treated. The major problem there is the absence of laboratories and clinics to make these measurements. The technology we developed, which is shrinking a laboratory onto a chip, biochips, disposable plastic devices which need very little in the way of detection instrumentation, are eminently suitable for that part of the world itself. So it's really suited to address the major need in developing world diseases like HIV/AIDS, malaria and TB.
Sensor technology is undergoing a revolution. As it becomes more sophisticated, it offers Irish investigators a unique perspective on human life, moment to moment, in every aspect of our lives. As a result of developments in this field, we have a more profound understanding of the chemical and biological processes of disease. The challenge now is to use the information that we get from this world of sensors to create new applications which will improve all our lives.